This presentation is in response to exam 3D, question number 2, concerning class switching. Immunoglobulin class switching is a mechanism that changes a B cell's production of antibodies from one isotype to another. Naive B cells can only express IgM and IgD isotypes. In these cells, the heavy chain immunoglobulin locus has previously undergone VDJ recombination that placed rearranged V, D, and J segments upstream of the C mu and C delta constant regions. Transcription initiated upstream of the rearranged VDJ segments will produce alternatively spliced mRNA transcripts that generate either IgM or IgD heavy chain proteins. After translation, the functional IgM and IgD proteins associate with light chain proteins and become expressed on the cell surface. So in this initial orientation, transcription of immunoglobulin arises only from the promoter located upstream of the rearranged VDJ and produces only IgM and IgD isotypes. The gene segments encoding the constant region for all of the remaining isotypes are located downstream of the C mu and C delta constant regions. With the exception of C delta, each of these other constant regions is preceded upstream by a sequence of DNA known as a switch region. Switch regions range in size from a few kilobases to nearly 10 kilobases. They're composed of hundreds of repeats of these G-rich, short DNA sequences. For example, the switch region located upstream of the C mu constant region is about 3 kilobytes in length, and it's composed of about 150 copies of this short nucleotide element, which itself contains 3 copies of the sequence GAGCT. So located upstream of each switch region is a DNA sequence that functions as a promoter called an I region promoter. Under appropriate circumstances, the I region promoter can be induced to initiate transcription by RNA polymerase II. Transcription begins just upstream of the switch region and continues through the constant region DNA segment. When this first occurs, messenger RNA that gets produced is known as a sterile transcript because it lacks an initiating methionine codon, and therefore it cannot be translated into a functional protein. So isotype switching is stimulated in the course of an immune response. So for example, an activated T follicular helper cell engages a B cell through linked recognition. The T cell induces expression of CD40 ligand and the production of cytokines, in this case interferon gamma. CD40 ligand engages a CD40 molecule on B cells that activates NF-kappa B, and interferon gamma activates STAT1 within the B cell. These transcription factors together to selectively induce the transcription of the I region promoter upstream of the C gamma 2A constant region. However, they do not induce transcription from other I region promoters. The different transcription factors activated by the cytokines determine the specificity of I region activation. So in this case, activation of STAT1 by interferon gamma leads to the activation of the I promoter region for C gamma 2A. Induction of transcription in the I region promoter opens up the DNA and allows an enzyme known as AID, activation-induced cytidine deaminase, to access the switch regions. So as RNA polymerase II transcribes the sterile transcript, the repetitive elements in the switch region induce a behavior in the polymerase that allows AID to deaminate single-stranded cytidine residues in the DNA and convert them randomly to uracils throughout the switch region. So through the action of the two other enzymes, these uracils are eventually converted into single-stranded nicks in the DNA on both of its strands. These processes also occur at the C-mu region or at any other switch regions actively transcribed in the B cell. These activities create multiple breaks in the DNA of the C-mu switch region, as well as the DNA of the switch region selectively activated by cytokines. Because the breaks have been produced in two distinct locations, at a great distance from one another, the double-strand break repair machinery joins the two switch regions. After the double-stranded DNA break repair machinery finishes its work, the damaged switch regions will have been spliced together at some random point, leaving remnants of each switch region on either side of the repair joint. So the rearranged VDJ segment is now placed a few kilobases upstream of a new constant region, in this case C gamma 2A. This now makes it possible for transcription initiated from the V region promoter to generate a transcript encoding the original variable region, but now in the context of a different constant region. When the messenger RNA is fully spliced, the intervening switch region sequences are removed, leaving only the segments encoding a fully functional C gamma 2A heavy chain. The B cell that has undergone isotype switching will begin to secrete antibody of this new isotype class.